Hello, this is Mrs. Ness-Deden, and today we are going to continue talking about how DNA gets passed on from one generation to the next. Last week we talked about how DNA codes for proteins and how mistakes in that DNA can cause genetic conditions if it affects the protein that's made. Hello, this is Mrs. Ness-Deden. We're going to continue our study of genetics by starting to talk this week about how DNA is passed from one generation to the next. If you remember, earlier this trimester, we learned about a process called mitosis, a process of cell division. Today, we're going to talk about a special type of cell division. It has a very similar name. It's called meiosis. And you can see a diagram of it on the screen right now. So the first thing I want you to do is look at this diagram and think about how is this similar to the process of cell division that we learned before, and what do you notice that's different? As we start talking about how DNA gets passed on, you have to remember a little bit, first of all, of how we store DNA in our cells. So DNA has that double helix shape that you see at the top of the screen, but it has to be kind of wound up around these histone proteins to create chromatin that exists in our cells. And then when cells divide, that chromatin wraps up even more tightly into these chromosomes. Remember, chromosomes are only visible during cell division. And so by the time we can see these chromosomes, the DNA has already copied itself. So a chromosome is actually two identical copies of that section of DNA. And we call those two copies sister chromatids. So kind of the left and the right side of that chromosome are identical copies of the same stretch of DNA. In human beings, we have 46 of these chromosomes in each cell in our bodies. Here you can see a picture of every chromosome in one human cell. This type of picture is called a karyotype, and it just shows you all of the chromosomes in a human cell. And you can see here that they are laid out in pairs. And so we use a word to describe that. We say that our cells are diploid. Our chromosomes exist in pairs. Another way to write that is we call that a cell being 2n. For humans, the number n would equal 23. We have 23 pairs of chromosomes. And so, of course, you can probably figure out we get one of each of these pairs from our mom and one of each of these pairs from our dad. And so how that information gets passed to us is what we're talking about today. The first 22 of these pairs are what are called autosomes. They're just kind of the normal chromosomes that are the same for everybody. And they are what we call homologous chromosomes. You can see the two chromosomes in a pair are about the same length, even if they're not totally laying in the same direction. They're about the same length. They also have the same pattern of those kind of dark and light bands. And what you can't see in these pictures is they hold the genetic information for the same traits. So maybe on chromosome 3 here, if there's a protein right or a gene right there that has something to do with your eye color, this would be the copy that you got from your dad. But there's also a gene right here on this chromosome in pair 3 that also has a gene for eye color. And this is the one you got from your mom. So you have two copies of every gene. And the two copies of the chromosome have the same information or have information for those same genetic traits. The very last pair down here you can see are labeled X and Y in this one. Those are called the sex chromosomes, and those are what determine whether you are biologically female if you have two X chromosomes, or biologically male. That's this one that you see right here that has an X and a Y. We'll talk a little bit more about those as well. They're still pieces of DNA that have genes that code for certain traits. We'll come back and talk about those a little bit later. Our question for today is, how do we get a copy from mom and a copy from dad? And that's through a special process of cell division called meiosis, because your mom's cells have 46 chromosomes, but she's only going to pass 23 of them on to her children. And so there has to be a type of cell division that takes those 46 chromosomes and cuts them in half so that only half of those chromosomes get passed on to her offspring. So you can see in this picture, these top two cells would represent the cell of a mom and the cell of a dad. And the process of meiosis is this cell division that cuts the number of chromosomes in half. So you can see here, instead of having two copies of each, there's just one copy of the blue chromosome and one of the yellow in each of these cells. Those cells are called gametes. So gametes are reproductive cells. In a female, they would be egg cells. In a male, they would be sperm cells. The word gamete also includes reproductive cells from other organisms like pollen cells from plants. Any type of cell whose job it is to carry genetic information to the next generation is called a gamete. And you can see here the gametes are what we call haploid. Instead of being diploid, di means two. Haploid, I see that word and I think half. It only has half the genetic information. It only contains one copy of each chromosome. Then, when an egg cell and sperm cell, if we're talking about humans, when they recombine, that process is called fertilization, which then results in an embryo, in a new organism that has the full set. In the case of humans, we would have 46 total chromosomes again here. So the process of meiosis helps to cut that number in half so that when those reproductive cells recombine through fertilization, we have an embryo, we have a new organism that has the correct number of chromosomes. Meiosis in a lot of ways is similar to the regular type of cell division, mitosis, that we learned about earlier. You may have noticed in the diagram the names of the phases are the same. We still have prophase, metaphase, anaphase, telophase. The way that the chromosomes kind of move to the middle and line up and then separate is basically the same, but there are some important differences that we're going to highlight today. One of the differences happens in the very first step, which is called prophase 1 in meiosis. You'll see why it's called prophase 1 in just a minute. During the very beginning, when the chromosomes first become visible and everything's kind of getting ready for cell division, one thing that happens here that didn't happen in regular cell division is that those homologous chromosomes pair up with each other to form what's called a tetrad. And while they're in that tetrad, so here's a tetrad right here, you can see these two chromosomes. One of these, the pink one, might be the one that this person got from their mother. The blue one might be what they got from their father. They pair up to form this tetrad, this group of 4 chromatids. And while they're together, this interesting process called crossing over happens. The non-sister chromatids, so one from the pink and one from the blue in this picture, actually kind of twist up and exchange some genetic information. You can see in the picture at the right, they have actually swapped some of the DNA between the chromatids in that tetrad. You'll see why that's really important here in just a minute as we look at what happens to these tetrads as they go through the rest of the process of meiosis. The other big difference is that in meiosis, in order to cut the chromosome number in half, the cell divides not once, but twice. So if this is our original cell, the first division is called meiosis 1. And during that division, during anaphase, the homologous chromosomes separate from each other. So they kind of paired up at the beginning, but then they separate. And you can still see those little bits where crossing over happened. And then these cells divide again. And during meiosis 2, the sister chromatids separate from each other. So the important thing here to notice, and the reason crossing over is so important, is by the end of this process, we have 4 cells instead of 2, and none of those 4 are genetically identical. Because of that crossing over that happened earlier, each of these has a unique combination of genetic material. That's one of the reasons that even though the same mom and dad might have many, many, many children together, every child is unique and different because they're getting a different combination of DNA, a different section of DNA from mom, a different part from dad. They still get half from each parent, but which half they get and kind of the combination of traits they get is different every single time. So to kind of summarize the big differences and the main things you need to remember about meiosis is, first of all, at that beginning stage, that crossing over happens. That's a really important place where we increase the amount of variability and differences that exist between individuals. The cells then divide twice, and that's what cuts that chromosome number in half. So that the end result we get from this whole process is we end up with 4 gametes, 4 reproductive cells that are haploid. They only have half the normal number of chromosomes, 23 instead of 46, and none of them are genetically identical. As we continue to talk about how the combination of genes that you get from your mom and your dad make the traits that you have, it'll be important to remember kind of where those genes came from, and this process of meiosis that makes each individual reproductive cell, every egg cell, every sperm cell, a little unique and different. We will continue talking about what happens with this DNA tomorrow. I hope you have a great rest of your day.